You know, every tissue in our body is this incredibly complex society of cells, and they all come from just a single ancestor. But how do we actually map out their family trees? How do we figure out who's related to whom to understand how we develop, or how diseases like cancer take hold? Well, today we're going to dive into a new method called MAESTER, which is giving scientists a way to trace these cellular family trees with a level of detail and at a scale that just wasn't possible before.
All right, so here's what we're going to cover. First, we'll talk about the fundamental challenge that researchers face in lineage tracing. Then, of course, we'll introduce MAESTER and get into the nitty-gritty of how it works. We'll see the clever ways the researchers proved it was reliable, look at a really powerful example of it being used on a patient sample, and then we'll wrap up by thinking about what this all means for biology moving forward.
Okay, so first things first, let's talk about the big problem that scientists have been grappling with for a long time. When you're trying to study tissues, you need to know how all the individual cells are related to each other. But the tools we've had have always forced this really difficult choice.
So, the central question is, how can you possibly tell which cells are related? The secret, it turns out, lies inside their mitochondria. Think about it like this. Every time a cell divides, tiny random typos or mutations pop up in its mitochondrial DNA. And these little typos get passed down to all the daughter cells. They act like unique inherited barcodes. So if you can read those barcodes, you can reconstruct a cell's entire family tree.
And this is where that difficult choice comes in. It's always been this classic trade-off. On one side, you've got these amazing high-throughput single-cell RNA-seq methods that let you look at thousands upon thousands of cells at once. The problem, the information you get about those mitochondrial barcodes is super sparse, almost useless. On the other side, you have methods that give you really rich, deep genetic data, but for only a tiny number of cells. It's like you could either have a blurry satellite photo of an entire city or a crystal clear, high-def photo of a single building. You just couldn't have both at the same time.
So this is where MAESTER enters the picture. It's a brand new technique that was specifically designed to bridge this gap, to finally give scientists the best of both worlds, that massive scale and the genetic depth. The name, MAESTER, is a bit of a mouthful, but it perfectly describes what it does. It stands for Mitochondrial Alteration Enrichment from Single-Cell Transcriptomes to Establish Relatedness. Basically, in simple terms, it takes the stuff you already made from a normal high-throughput experiment and just boosts the signal from the mitochondria. It makes those genetic barcodes loud and clear. Now, what makes this a real game changer for the research community is just how accessible it is. MAESTER isn't some niche technique that requires crazy new equipment. It's designed to work with the platforms that tons of labs are already using every day, like 10x Genomics, seqWell, and DropSeq. This means it can be adopted pretty much anywhere. All right, so let's get into the nuts and bolts. How does this thing actually work? The magic of MAESTER really comes down to two key parts: a really clever tweak in the lab protocol and a powerful piece of software to make sense of the data. The process itself is surprisingly elegant. You literally start with the amplified cDNA that you already have from your standard experiment, so no need to go back to square one. Then you use a special set of primers and a PCR reaction that acts like a searchlight, finding and making millions of copies of only the mitochondrial transcripts. You sequence that, and then the MAGCat software takes over. It just piggybacks right onto the existing workflow, which is incredibly efficient. And the result of this little tweak? It's pretty staggering. MAESTER boosts the coverage of the mitochondrial genome by more than 50-fold. So what does that actually mean? It means for any given cell, you go from having just a few scattered, unreliable pieces of information to having a deep, comprehensive readout of its mitochondrial DNA. And that depth is what lets you spot those barcode mutations with real confidence. But you know, getting a ton of data isn't the whole story. You have to be sure it's good data, right? That's where the software, the MegaGat toolkit, is absolutely crucial. Think of it like this. Sequencing is kind of like making thousands of photocopies. Some of those copies are going to have smudges or errors. Well, MegaGat uses something called Unique Molecular Identifiers, or UMIs, to find all the copies that came from the same original molecule. It then stacks them up, compares them, and builds a perfect consensus, filtering out all the noise and errors. This way, you know the mutations you're seeing are real. Okay, so this all sounds great in theory, but for any new method, the big question is, does it actually work? As you'd expect, the researchers didn't just take their own word for it. They put MAESTER through some seriously rigorous tests to prove it delivers.
For one of their key tests, they did this really smart experiment where they just mixed two different types of human cells together. When they looked at the data from the standard method, the mitochondrial coverage was, well, it was basically nothing. But after they applied the MAESTER protocol, the coverage just skyrocketed to over 130-fold. I mean, that's not just a small bump, it's a monumental leap.
So, the million-dollar question: did that massive boost in coverage actually help them tell the cells apart? The answer was an overwhelming yes. The family trees they built using the master barcodes matched the cell identities from the standard gene expression data with 100% accuracy. It was a perfect match. It cleanly separated the two cell populations, no problem.
And they didn't stop there. In another really robust validation, they used a fancy technique called DOGMA-seq, which let them measure both the mitochondrial DNA and the RNA from the exact same cell. They used the DNA as the gold standard, the absolute truth. So how did MAESTER do when they looked at just the RNA? It was incredibly accurate, successfully recovering over 94% of those true gold standard variants. This proves the method is not just finding things, it's finding the right things.
Okay, proving a method works on clean cell lines in a dish is one thing. But what happens when you throw it at the real world, at a messy, complex sample from an actual patient? That's the ultimate test, right? Well, the researchers analyzed a bone marrow sample from a patient with a condition called clonal hematopoiesis, where different families of stem cells, or clones, start to take over.
And this is where the power of MAESTER just really shines. From this single patient, they analyzed almost 10,000 cells and identified 23 distinct clonal families. And what's more, they found that these families were behaving differently. Some clones were biased, making way more T cells, while others were churning out myeloid cells. And then, this is the really critical part, they were able to link 2 of those myeloid-biased clones directly to known cancer-driving mutations. This is the kind of disease-relevant biology that would have been completely invisible without this technology.
The authors themselves put it perfectly in their paper. They said, MAESTER enables discoveries that are not possible with each method alone. It's not just about adding another piece of data. It's about the synergy. By combining it with standard single-cell analysis, you get this incredibly rich, multi-layered view of what's really going on inside a tissue.
So, let's zoom out. What's the big picture here? What does a tool like MAESTER really mean for the scientific community, and where do we go from here?
The big significance is that MAESTER basically democratizes mitochondrial lineage tracing. By making it work with common lab equipment, it puts this incredibly powerful approach into the hands of way more scientists. You can even go back and use it on old samples you have stored in the freezer. But like any tool, it's not a magic bullet. It's important to know its limitations. Because it relies on those naturally occurring mutations, it's not going to track every single cell division. It's really best for finding those bigger family relationships between cell populations that have had some time to diverge.
So at the end of the day, MAESTER gives us a powerful new lens to look at really complex biology. It's opening the door for scientists to reanalyze old data sets and to design totally new experiments to explore these clonal family trees in development, in aging, and across a whole range of diseases. And it leaves us with a really exciting question to think about. What hidden cellular dynamics, what secret family histories that are playing out in our bodies right now will this new tool help us discover next?